Oh, what's up? I'm here again. I know you want to hear from me. Okay, and you're going to hear from me anyway. So let's talk about mitochondria today. Mitochondria. Okay. Before I get graphics for these videos, I'm going to just get good with the talking and explaining of things so it makes sense to people. Eventually, I'm going to get the graphics and make them shiny and flashy and totally worth YouTube advertising revenue. Okay, but not right now. I know you got your like expert biologists out there that are all about that YouTube advertising revenue, and I'm not there right yet, but eventually I'll get there. No, I'm not even about that. Okay, I'm here about talking about eternal life. All right, instead of rambling, okay, let's learn today. So I want to talk to you. Let's bring up JS Paint. I've given up. I mean, you know what? I don't need to use JS Paint solely, but I'm just going to do that because it's faster right now. I'll research some other paint application I want to use for doing diagrams. Wow, cool, that applet. It's definitely an applet, right? Anyone remember Java applets from back in the day? You probably don't, but applets were used in the 90s as a way of extending functionality to JavaScript. Yeah, I'm also going to talk about a few CS things here and there, but you would embed them in websites and they could do all sorts of things like cool diagrams, whatever. Anyway, right now they can, in this case, this applet still provides functionality to this web page in the form of a paint app. You could do something like this in JavaScript. In fact, I've seen some really good paint apps in JavaScript on a website, but did I also mention that I think I'm going to mention it right now. I'm going to do a bunch of different topics interspersed together while I talk about biology and mitochondria and whatever. And all eternal life stuff eventually kind of flows together. But let's talk about mitochondria. What are mitochondria? So in school, you learned that mitochondria are these jelly beanish like organelles in a cell that look kind of like this. Okay, that's the historical diagram that most middle school textbooks give to mitochondria. Okay. Oh man, mask is falling off. And what they do is essentially they are, as people like to say, the powerhouse of the cell. All right. Okay. So what does that mean exactly? That means that mitochondria are responsible for the production of ATP, okay? And ATP, remember, if I haven't gone over it before, at some point in the future when I do a proper series of lectures, ATP stands for Adenosine Triphosphate, okay? What does that mean? Adenosine triphosphate. So adenosine is a nucleotide, right? Triphosphate means that there's 3 phosphates attached to this nucleotide. So what is a nucleotide? See, that's the thing. When I talk about one thing, I've got to get into everything else. But I'm going to do it like the experts and say that we're going to talk about ATP more in some other video. But let's just quickly go into what ATP is. Okay, what is ATP? Since mitochondria produce ATP, we're going to talk a little bit about that. So ATP is a nucleotide. And what is a nucleotide? So I'm getting my molecular chops going, right? And a nucleotide is basically something that composes DNA, right? Your DNA is composed of several nucleotides. So nucleotide is basically your standard 5-ring ribose sugar, okay? And I say ribose because this is a ribose, okay? And what is a ribose? A ribose is a sugar that has an oxygen in its ring, its constituent sugar ring, and several hydroxyl groups surrounding it. And it's attached to the adenosine group. Okay, so this is adenosine. Now I think I forgot the structure of adenosine, so probably not good to go super into this, right? Otherwise I'll confuse people. But adenosine is basically it's a molecule that's composed of two rings, right? Two carbon rings. These rings are carbons, by the way. Okay, I haven't gone into organic chemistry, and I don't know how deep I should go into their organic chemistry, but NH. So this is adenosine, okay, and it's basically this is an amino acid, right? Common amino acid attached to a ribose sugar, which is then attached to a phosphate. Okay, so what is a phosphate? A phosphate is a phosphorus attached to oxygen. Oh man, I'm getting this totally wrong. But and this is remember, this is a triphosphate. Okay, so you have three, and a phosphate is attached to three oxygens. Sorry, phosphate is attached to four oxygens. Phosphate group is attached to three oxygens. Okay, so you have this. So let's just look up phosphates. Okay, we're going to look at all the components of ATP because we got to know what the mitochondria produces. ATP. Adenosine triphosphate. Okay, so this is the structure of a phosphate. Let's zoom in a little. Okay. Okay, you see the phosphates are these phosphoruses attached to these oxygens on the top, which it's an oxygen with phosphorus in the center attached to each covalently attached to the phosphorus with hydroxyl groups kind of filling out the oxygen on the ends, right? So you have hydroxyl, hydroxyl, hydroxyl. So you can think of the phosphate just looking at like this, phosphate. We'll bring it up in another tab so you can compare. Phosphate. This is good to remember because if you're ever asked to find out what a phosphate group is, or what the adenosine triphosphate is composed of, which is something you're going to do in some point in your biology career, you should know. Phosphate is this oxygen, 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 one double bonded oxygen, three single bonded oxygens. This is one that's going out of the screen and one that's coming into the screen, right? So this is a tetrahedral molecular structure that the phosphate is in chemistry. A phosphate is an anion salt functional group or ester derived from a phosphoric acid. Okay, and what is a phosphoric acid? Phosphoric acid is basically where you have the H's here, the hydrogens attached to each of these oxygens. The one that's double bonded is stable, it doesn't need a hydrogen attached, but a phosphate is the anion of it's the complete anion of phosphoric acid. And these are the different types of hydrogen phosphates you have. Hydrogen phosphate has one H, just a phosphate anion by itself has no hydrogens bonded to it. Dihydrogen has two hydrogens, phosphate has two hydrogens, and phosphoric acid has three hydrogens, and it's in its sense is the stable version of the molecule because it's got the hydrogens attached. But phosphoric acid is I believe a weak acid, so it has a strong a conjugate base. So let's just go check how strong general phosphoric oxoacid, right, that's the general term for it, which each phosphorus atom is in the oxidation state +5, is bonded to 4 oxygen atoms, one of them through a double bond, so +5, so that's 1, 2, 3, 4. The oxidation state is stable, right, for this centralized phosphorus, and arranges a tetrahedron. So let's not go into the weakness or strength of phosphoric acid because we just wanted to show you what the phosphate was. So now that you have the phosphate, going back to adenosine triphosphate, you have these hydrogen phosphate groups attached to each other, right? This is this bond here attaches the phosphorus, so it's tri because you have 3 phosphates attached here, and then you have that attached to what this is called the 5' carbon. 5 because you have 1, 2, 3, 4, 5. Okay, this is the 5' carbon if you number all the carbons, and down here is the 3' carbon. In DNA, you'll see this also attached to phosphates, which form a single phosphate, which will form a DNA, the phosphate backbone of DNA. Anyway, and then this is the amino acid part. This part I'm circling right here, which contains the purine. I believe this is a purine called adenosine. Okay, which is the amino acid, and in DNA, it's the amino acid that codes for A. Right, A for adenosine, or adenine, adenine, right? Adenosine comes from this sugar here, right, which is a pentose sugar. So pentose sugar connected to adenine makes an adenosine. This bond is important because this is called a glycosidic link, an N-glycosidic linkage. Okay, you don't need to go that deep into it. Okay, but anyway, so ATP is important because, in short, this phosphate will break off from the adenosine triphosphate, and this is a high energy phosphate, so that breaking off releases energy. It's an exothermic reaction, and that energy is then used in propelling other reactions throughout your cells, right? It's very important because everything uses ATP. And converting it to ADP, or oxidative phosphorylation, as you can say. And that oxidative phosphorylation process is the primary mechanism by which cellular functions actuate, right? Okay, now that we know what ATP is, right, let's go back to this. Mitochondria produce ATP. They produce ATP through cellular respiration. Okay, cellular respiration is a fairly complex process, and you've probably learned it at some point in high school biology, and you probably forgot all the steps, but let's not talk about it here. So we have mitochondria, this card. I'm going to draw this elementary school diagram of mitochondria again. So what do we have here? Mitochondria is a membrane-bound organelle. So once again, phospholipid bilayer. I was talking about yesterday about the phospholipid bilayers of mitochondria, of any membrane-bound organelle, like vacuoles, for instance, or the endoplasmic reticulum, the nucleus, the smooth endoplasmic reticulum, the rough endoplasmic reticulum, the Golgi body, lysosomes, peroxisomes, vacuoles, all these membrane-bound organelles, right? And even the transport packages that these membrane-bound organelles create in order to transport proteins to other membrane-bound organelles or to the cell membrane itself, right? So the mitochondria is a double membrane-bound organelle. Mitochondria, double membrane-bound. And that's important because the mitochondrial membrane, the intermembrane space of the mitochondria is very, very important. Okay, so basically, what does this membrane, the outer mitochondrial membrane, do? What's special about it? Because there is a difference between the inner and the outer. The outer membrane contains holes. Okay, or more specifically, in a more microbiological level, biochemical level, it contains proteins called porins. Okay, porins. So what are porins? Porins are responsible for passive transport of molecules, small molecules into cells. Okay, they're like pores, right, on your skin. But pores in your skin, they let sweat out normally, right? But these let micromolecules, small enough molecules, travel in and out. Okay, so they're considered permeable to small molecules. Important to remember, permeable to small molecules. Okay, why that has to do with cellular respiration. We're not going to get into all of that, but again, I can put small molecules here, right? But we have to remember that it won't allow larger molecules in. Okay, that's very important because these small molecules, right, are both inside and outside. Because this is permeable through the porins, right, this concentration of molecules that are outside and inside will be the same, okay? So that's kind of like how osmosis works, right? You have a potential equalizing function created by a semi-permeable, in this case, permeable membrane, okay? This inner phospholipid bilayer, oh, by the way, if we zoom in on this, remember, I think I might have shown you videos. You have the phospholipid bilayer, right, which contains your phosphates and your phospholipids, right? And then it contains a huge porin protein, okay? So this porin protein is really interesting. I mean, basically it functions as an open channel for small molecules, but it prevents larger molecules from getting in. But let's take a look, because I really want to show you what a porin protein looks like. Porin protein. A lot of people probably, especially if you're in high school, take the complexity of proteins to be, for granted, right? You'll be like, oh, this is a protein, that's a protein. But they don't really go into this too much as they should in high school biology, but this is a porin protein, okay? So these are examples of porin proteins. Porin proteins are in your nucleus, they're on your mitochondria, they're in a bunch of places. Porin proteins are also on your cell membrane, your outer cell membrane, okay? And basically this example, this here, is a barrel protein. Right. Why is it a barrel? Because it looks like this. What are these? All right. I'm going to keep going over and over these again and again. But this is what you can call a beta barrel, right? Beta because these crisscrossing thingies here, the way these proteins are organized is in a tertiary protein structure called, or secondary protein structure called beta sheets, right? And then basically you have several of these beta sheets all linked together from the same protein, right? And thus they're called beta. This is called a beta barrel protein, right? And it's very simple. Like the funny things about proteins, right? Proteins are molecules, right? That are composed of amino acids that are linked by peptide bonds, okay? So it's strange the way complexity works in biology, or in life in general, or in the world, in the universe, in physics. Your amino acids are quite complex on the molecular level internally, like in terms of the interactions of the different atoms. But when you get out of the molecular level, your amino acids basically link together to form proteins, right? Via peptide bonds. Okay, peptide bonds are bonds that link amino acids together. You can take a look here. We'll take a look at a peptide bond. Peptide bond. Okay. A peptide bond. Let's see if there's a good image of a peptide bond. Basically, these are two amino acids linked together. I don't know which amino acids these are off the top of my head. But basically, a protein is formed when several amino acids are linked together. Okay. So you can imagine that it's a protein. That beta barrel is composed of several amino acids. Like they took out the actual individual amino acids here because it would be a pain to draw them all. But you can imagine that a beta barrel porin protein will look something like this. These are beta sheets, right? And they call, again, they're sheets because the space between these proteins, these individual strands, is hydrogen bonded. Okay, that's why you don't see a link like that. The proteins that are single continuous lines are covalently bonded amino acids, and there's no point in showing them because their structure usually is not going to be the one in question in a protein or protein interactions or whatever. It's usually the hydrogen bonding structure, the weaker bonds which keep the tertiary structure together, or secondary structures together within a protein, okay? So basically, you have a bunch of these beta sheets that are linked together like a coil, And this is what I was saying, the complexity is strange, right? I mean, amino acids linked together are not very complicated. They form a linear series of molecules, which is basically like a fence. Physically, it acts like a very microscopic fence as well. But on a larger scale, right, these things combine together through interlinked hydrogen bonds, which is very interesting because these hydrogen bonds, the science behind hydrogen bonding is quite complex. But then if you take it a higher level, even higher, this is basically a barrel-shaped protein. Now, why this is a barrel-shaped protein, which is very convenient because it's supposed to restrict the flow of things in, right? The diameter of this barrel restricts the size of the molecule that can enter the mitochondrial wall, right? Phospholipid bilayer. And all this complexity really kind of fluctuates, right? Because at that level, it's not that complex. A barrel is just a round tube, okay? All right, so this thing, basically, you would think this protein functions very mechanically, as proteins often do. We've gone into the motor proteins in a previous video, and I showed you. Actually, let's do that again, because I like showing motor proteins. Motor protein animation. The motor protein animation, these are freaking just, like if you look at the motor proteins, these motor proteins are, it's quite a beautiful kinesin moving. This is a protein that moves, but this is like pretty much very close to how kinesin actually moves, right? It's, I think, bringing a virus along with it across this microtubule, but this kinesin is, these are protein domains, and these complex, very, 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 very thousand molecule, thousand amino acid complexes basically are something that evolves to act like legs, and legs are at a fundamental level very important to motion, right? So anyway, I just thought I'd go a little into that. I probably went too much into that, so let's go back to porins. Okay, porin. So porin, Wikipedia is my friend. Wiki is great. Protein is a transmembrane protein. Okay, so it's basically these beta barrels, and bacteria, gram-negative porin means bacteria have them as well, and thus the mitochondria have porin proteins. Okay, so let's go to the mitochondria. Back to the mitochondria, outer membrane, which the mitochondria were bacteria at some point, according to the endosymbiotic theory, anaerobic bacteria. So their porin proteins probably don't differ much from their ancestors' porin proteins. Okay, so at some point I'll make a proper mitochondrial video instead of doing this free-form thing. But I kind of wanted to get this going. So inside the mitochondrial's inner membrane, which is folded like this into structures that are called cristae, right? It's not folded exactly like this. According to newer research, the cristae don't look like this, but the cristae look like some really complicated cavernous thing that twists and bends on itself. So the inner membrane isn't actually, it's not a nice little lumpy, snaky thing. It's actually quite a, hold up cavernous item with a structure within the mitochondria. And inside this inner membrane is where most of the action of cellular respiration happens, where ATP is formed and stuff like that. So basically, cellular respiration occurs here, and then the ATP leave the mitochondria. That process, still something I'm going to have to find a good way of explaining to you. But at a fundamental level, that's all I want to talk about for right now. We can talk about endosymbiotic theory later. I touched on that a bit because the mitochondria are essentially, mitochondria are anaerobic bacteria, which were absorbed by our eukaryotic ancestors' cells about 1.5 billion years ago. I think it was about 3.8 billion years ago, or 1.5 billion, I forgot. It's in the fossil record somewhere. They were absorbed, and they, instead of being eaten by those eukaryotes, they were, permanently lodged in there, and they sort of decided that they could live together because the eukaryotes, these mitochondria needed to hide, right? They needed to hide because oxygen actually caused damage to the mitochondria. They're anaerobic, and anaerobic bacteria, and many of them are damaged by oxygen. And at that point, oxygen was in the atmosphere. So the eukaryotic cells love oxygen, right? But they need energy to function. So these mitochondria produce ATP. These guys need ATP. These eukaryotic cells love eukaryotic because they're your nucleus, your true nucleus. Eukaryotic cells love oxygen, so they absorb oxygen, but they don't allow the oxygen to get to the mitochondria to destroy them. So the eukaryotic cells protect the mitochondria, and thus you have a symbiotic relationship where the mitochondria provide ATP to the eukaryotic cells to do their functions, and the eukaryotic cells prevent the mitochondria from being degraded by or oxidized by oxygen. So what will happen is that the eukaryotic cells, when they divide, they create copies of the mitochondria along with them. It's actually quite an interesting theory, and no time to get into it now because I'm way over for this video. So anyway, peace.